So good morning friends, my name is Manoj Kumar Yadav. I am a professor of Biotechnology at Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut, and today I will deliver a lecture on entrepreneurship opportunities through plant tissue culture technology, and this system contains my lecture will consist of 4 parts. First is the basic plant tissue culture technology and basic concepts of the plant tissue culture technology. Then second is the laboratory organization, how plant tissue culture technology it consists of. So it consists of different parts, and then how entrepreneurship may be developed using plant tissue culture technology. So different cost benefit ratio is also discussed in this lecture. Then we will discuss about the successful examples of this technology. So we will discuss, and we are going next in this process. Topic of discussion is the entrepreneurship opportunities through plant tissue culture technology, and this is a very interesting topic. So we will start this section, or the complete section will divide into 4 parts. So we will discuss first about the plant tissue culture, the basic techniques, and what is actually the plant tissue culture is, and then plant tissue culture laboratory organization, how laboratory, if someone want to establish, so what different type of parts required or infrastructure required for to develop a plant tissue culture laboratory, and then how plant tissue culture laboratory or technology may be used as an entrepreneur entity. So, and then successful examples in India about plant tissue culture, how this country is running different types of successful plant tissue culture units, and then we'll again discuss what is actually plant tissue culture. So it is also known as in vitro culture. In vitro means glass. Glass means under artificial conditions. So that is why this is known as plant tissue culture. Means we can culture any plant parts, that is known as explants. Means any plant part, that may be root, shoot, leaves, anther, pollen, or microspore, and we culture it on a artificial media, that is a Murasic and media, and it is in short is known as MS media, and all the operations were done or completed under controlled environmental conditions. Means environmental conditions, they include lights, say optimal light provided, that is artificial lights, through tube lights or some other power lights. Then we have to maintain the humidity and the temperature. So temperature, humidity, we should keep 75% humidity, not more than that, and temperature, it should be maximum 25 degree Celsius, not beyond that. And then next important part, although there are many other terms like explants, in vitro culture, and cloning, clone means any plant part. If we develop any plant that is morphologically or genetically identical, and all plants they are similar, in that terms they are known as clones, and the process of making clones, that is known as the cloning. So cloning may be done in plants and also in microorganisms, animals, and even human also, but that is a different section for discussion, especially in animals and humans. So here we are dealing with the plant tissue. So plant tissues, they are having the capacity of or phenomena of totipotency. Totipotency means any plant part can develop a complete plant, but none of other organisms they can develop, especially in human and animals, they do not have capacity of totipotency. That is why we cannot develop any complete plant animal parts from a single cell due to lack of totipotency there. So explants, that is a crucial for any tissue culture technology or tissue culture systems. So here physiology is an important criteria to select explants, and season, in which season we have collected that explant, that is also matters. Then size and location of explant, which kind of, which how long or what is the diameter of that uh, explant is there, so that it depends on the success. If explant is large, so not, but it should not be too large, it should be from 1.1 mm to 0.5 mm in size, but beyond that 10 mm or 2 mm 20 mm, it is suitable. And from which location we collected the explants, it depends the success. The quality of the source explant, whatever plant we are choosing, what is the source of that plant, means whether that is healthy or unhealthy or disease. So we should select a healthy plant parts, or so that is also crucial. And so it helps to improve the culture. Then we will discuss now about the plant tissue culture media. So I already have told that plant tissue culture media, that is, this is the MS media, and it is the most commonly and accepted media in plant tissue culture industry. And it was again, uh, I already have discussed that two Japanese scientists, they have devised this media 1962, although other types of media also exist, like N5 and many other types of media are there, and most of them, they contains major constituents like major salts or minor salts. So major salts are carbon, hydrogen, oxygen, like that, and like micro salts, they are like zinc, molybdenum, iron, they are the micro salts. And each and every plants, they need 17 to 18 uh, elements or salts and their combinations for a proper growth. So if a plant, we give them optimum concentration of the salts, then plant grow very well under artificial conditions also. Then we supplement the media with sucrose or sugar source like sucrose, glucose, and inositol. And here vitamins are also used. Then some other natural supplements are also used, like coconut milk, that is rich with many minerals and vitamins and many other growth factors. So in the earlier time, coconut milk were used, but nowadays no one used, or rarely it is used for plant tissue culture. Then supporting media, that is inert in nature, so also used as a solidifying agent. So agar agar is used as a solidifying agent. Sometimes gerlite may be used, but agar agar is the most commonly used inert supporting material in plant tissue culture. But in many other cultures like sugarcane, we use liquid media. So there is no need to add the agar in media in that case. Then growth regulators, so they are also known as phytohormones. So they regulate the growth, and there are different classifications like auxins, cytokinins, and gibberellins. They are mainly used in commercial plant tissue culture, although many other growth hormones are also used. Then organic compounds are used as a carbon source. Sucrose is most commonly used, while glucose is mainly used in haploid culture or anther culture. Vitamins are also used here in plant tissue culture, but in field plants we never feed them with vitamins. So it is very different here, and very interesting that plants, they consume vitamins here in the media. So thiamine and pyridoxine and many other vitamin B complex part of the vitamins we use here. So, and they are the essential part and work as a coenzyme in citric acid cycle and many other metabolic pathways. We also add sometimes adenine, that is a part of DNA or RNA, and inositol, that is a part of B complex. Still, other organic acid may also used, like organic acid, citric acid, and that is used again in citric acid cycles. And the phenolic compounds also used, like fluoroglucinol. It stimulates rooting in shoot sections during culture. And then sometimes many woody plants, they contains loads of phenolic compounds. So that creates problem, and browning occurs during culture, and plants or plantlets, they do not grow very well. So subculturing is needed, and in that case, in such situations, such circumstances, inert charcoal or activated charcoal is used as a detoxifying agent, and it removes or it reduces the intensity of those phenolic compounds, and periodic subculturing is used. So it is used in a very small amount, and it cannot be it or it should not be reused in the media. Then these are the again plant growth regulators. So auxins, it contains the like NAA, IAA, and 2,4-D, 2,4-YT. So of them, NAA sometimes or IAA used. Indole acetic acid, that is the natural auxins, while cytokinins, it induces shooting during culture. So there are different type of cytokinins like benzyl aminopurine, BAP, and GRT also. It is obtained from GMAs, but BAP is used because it is cheaper and synthetic. And gibberellins, that is used for cell enlargement, and GA3 is used sometimes, and abscisic acid, which is also known as senescence hormone or plant stress hormones. So it is used. It is not used in the micropropagation industry, but it is used in the research purpose for regeneration purpose. Then ethylene, that is known as ripening hormone, but it is mainly used for ripening process, and it is not good for but in micropropagation industry, and it is not considered good. So now we will discuss what are the different parts of the plant tissue culture laboratory. So how a plant tissue culture laboratory is organized. It consists of different parts like media preparation rooms. So it is a building, the media inoculation room, washing area, culture room, and all the things, they should be well contains air conditioners, and all the operations they have maintained under a optimum temperature regimes, and it should not be more than 25 degrees centigrade at any cost. And then we need hardening unit or net house, even a field, so that we acclimatize the plants, and then they get acclimatized and hardened, and they grow further happily. Then we any unit or any system that also need a unit office, because that is the governing body of that system, and then a small storage also needed to put the expensive chemicals, although many rare chemicals and like hormones, they stored at a low temperature, and refrigerators of different temperature range are required. So these are the equipments, and this is the list of the equipments which are used. They are the major equipments which are used for a plant tissue culture laboratory. So these are the laminar airflow. So here horizontal laminar airflow is used, although and another type of laminar is exists, that is the vertical, and that is used for the microbiological operations. And refrigerators, different type of refrigerators from -4 degree to -20, even sometimes -80 degree centigrade deep freeze are also used. And culture bottles, They are made up of glass and autoclavable caps and different type of other glasswares are also used throughout the operation of the for making culture media, and that media is utilized with the help of either pressure cooker or with the help of uh, autoclave, hot air oven, and microwave are also used. Many other, because in this system pure water is used. That water should not contain any kind of salts or impurities. So that is water purification system or distillation unit is also required uh, in a plant tissue culture unit. Then weighing balance, pH meter, vortex shakers, microscope, and stereoium are also required if someone want to take pictures or to do some research. So microscope are also used. Even vacuum pump and growth chambers are also used to grow the hardened plants sometimes, or making them acclimatize after growing them in the on the culture bottles. And then humidifier also used. The humidifier also help to maintain the humidity in the culture room. Then these are the other hot plates and other requirements. Many other minor requirements also be there, like scissors, forceps, and needles, and data collection area and data book maybe also required. Even every person nowadays they are using uh, good quality of good high capacity computers and printers. So majority of the commercial plant tissue culture units in India, they are situated in many parts of the India, like southern, central, or western parts, and the production capacity that is actually needed is about 500 million plants, while they are producing at present the 300 million plants every year, and estimated market size in 2024, that is around 800 crores. And India, that is why India is focusing on the producing banana, sugarcane, ornamentals. There are different type of ornamentals like carnation, strawberry, antirrhinum, lilium, and orchids. And stevia, gerbera species, potato tubers are also producing in many research laboratories. And these tubers, they are used as a microtubers, and for potato seeds, even some synthetic seeds are also used. And medicinal plants, turmeric, apple, banana, and many uh, woody plants like teak and pomegranates. So these plants may be obtained from plant tissue culture. So now question comes, why micropropagation? What is the need of micropropagation? Because it is a very good technique, and it is a good alternative for vegetative propagation, which multiply the plants many times in a short period of time. That is why large number of identical plants, that they are known as clones, may be achieved from a single tissue within a short period of time. A multiplication cycle is very short, and each cycle resulted in a logarithmic increase in the number of plantlets. So a huge number of plants may be achieved or may be obtained in short period of time. So this is the beauty of this technology, and germ plant may be maintained for many years through this technology, and all plants or germ plant, that is the free from bacteria, fungus, and viruses, and that is why plants are good in quality. Millions or thousands of plants can be achieved in bottles simultaneously, and this is a further a cost-effective technology through which new lines may be developed and propagated. Here, one more advantage is that genetic uniformity may be maintained. Then this technology is independent of the season, so we can grow any plant from any part of plant throughout the year. So there is no restriction of the seasons. So it is free from the seasons, and throughout the year we can grow So, dear friends, now question comes to what type of plant tissue culture facilities may be there. So, there are in general four type of plant tissue culture facilities may be established. One is the research purpose, and that is established by a university or any institute. So they are mainly used for research purpose, and sometimes they are commercially utilized nowadays. As the time proceeds, then small scale industry. So it is for a beginner as a startup facility. So if someone that is a new and want to establish a want to start a startup, so he start from a small scale and then medium scale. If someone is successful through a small scale industry, then he can upgrade their laboratory or their facility in a medium scale, and then it may be upgraded to the large scale for further for a, as a entrepreneurships. So it is a good industry. So these are the plants which are used commercially as an entrepreneur. So if someone starts from a small scale industry, so through a small scale, if suppose someone uh, want to produce 3 lakhs plantlets every year, and the plants types they are the maybe banana, sugarcane, ginger, medicinal plants that includes chlorophytum, bolivianum that is known as saped musli, aloe barbadensis, and ornamental plants such as carnation that is known as Dianthus caryophyllus, tuberose orchids, vanilla, and maybe some other, but they are the tentative. They are the 5 groups one should produce through small scale industry. But before establishing a industry, one should know that location is very important to establish a plant tissue culture unit or a successful business. Otherwise, business may fail. That is why a plant tissue culture lab should be located in a place where moderate climatic conditions exist and having uninterrupted supply of water and power. So power is also must, and 24 power supply, because there are lots of operations for light and controlled conditions of temperature. So AC are also running 24 hours without any fail. That is why these conditions may prevails, and we should or one should kept in mind these things. Tissue culture operations have to be carried out under controlled conditions of temperature. So extreme climatic conditions are not suited for production, and if someone establishes in extreme conditions like such a very high hot climate or low temperature regimes, then cost of cultivation may increase, or sometimes unit may fail. So one must consider this point very sincerely before starting the startup or doing any operation. So now we will discuss what is the economics for of starting plant tissue culture as a startup. Although it is a good business, so economics we will discuss. So suppose 3 plants. This is the estimate for 3 lakhs plantlets per annum from a small industry or a small business of this facility. So micropropagation business can be started as an entrepreneur with a small investment by setting up hardening unit to start with a small labor. So such entrepreneurs may procure primarily hardened tissue culture plantlets through a well-established lab, and then that should be sold to the farmers of the nearby area, and from there he or she may get income, and thereafter he should think to develop his own uh, unit, because it requires lots of money. So after establishing the market, a full-fledged micropropagation unit could be set up, and this is the estimate of the project. So it required fixed asset, and so land development and land and site developments, greenhouse, electrical fittings, furniture and fixtures, and total cost of these assets there may be around 100 lakhs for a small unit. If someone is having own land, then it is also good, and it may reduce further 40 or 50 lakhs, at least 40 lakhs from these 100 lakhs. So it may help for an entrepreneur. Then recurring expenses per year. So they are the like raw material, manpower, and utilities and contingencies. So total amount of this section or recurring expenses per year is 38 lakhs, and capital investment total, if we add it section 1A or B, that is fixed and recurring expenses, that is 138 lakhs, and it is a large amount for a common man. And then this is the financial arrangement. So one should procure or should arrange 100 lakhs or 1 crore rupees from through debt, or then he can pay 38 lakhs from his own expenses, or he may arrange from somewhere else. So this is the ratio, and this is the, suppose if he is getting loan, so at least 12% interest rate is included here. So rate of establishment or cost of the unit may increase or decrease, depends on the inflation and conditions of that area, and cost of the land and other inputs. So production cost, this is the recurring cost, 38 lakhs. Then depreciation, maybe if we take it 10%, then interest 12%, then total cost of the production is 46.2 lakhs, and annual turnover is, if this laboratory is producing 3 lakhs, then, and selling price per plant is 12 rupees per plants minimum. So total turnover per year, this is the 30 lakhs. So, and so probably in three years, a person will get free from all the expenses. Then mitigating the risk of the commercial plant tissue culture. There are lots of risks if in this industry. So utilization of plant tissue culture for commercial production is limited by two major risks: spread the disease caused by viruses and other pathogens. So, and risk of the distribution of the inferior micropipette plants is a major concern in this industry. So in order to prevent these risks, effective testing or indexing system should be adopted before culturing the plants or before hardening the plants. And then a standard procedure should be adopted, such as carefully selection of mother plants and ensuring the establishment of virus-free culture through indexing. So virus-free indexing may be done by ELISA technique, that is again very expensive. Then question, now we will turn, who can develop this facility? Although this is a bit expensive entrepreneurship system, but if someone is skilled, and if anyone who is interested and who is graduate, postgraduate, or having doctorate degree, or maybe MBA degree, so he or she or group of persons may start this business, and they should take training from some related entrepreneurial institute they are existing in India. So there are so many institutes which are exist in India. So they are specialized entrepreneurship development agencies, and they provide training, and they train the persons, and they tell the skills and the systems how to establish a industry, or not only plant tissue culture but any kind of industry. So this is the list of the institutes. So if someone are thinking to establish this industry, so they should contact these institutes, and they may get training from this, and resource of these institutes in detail it is giving here in this slide. So government is focusing on the startup industry. So government it is a government is playing important role in developing entrepreneurship, and government develop industries in rural and backward areas by giving various facilities with the objective of balances region development. And government is setting programs to help the entrepreneurs in the field of technology, finance, market, and entrepreneurial development, so that they help to accelerate and adopt the change in industrial development. So there are so many funding agencies. They may finance for any business, even for plant tissue culture units. So if someone is thinking to set up this entrepreneurship, so he or she may get finance. So major finance, if someone is thinking in this direction, so banks are also available. So many banks, they may provide loans. Besides that, Ministry of Agriculture and Agriculture and Processed Food Product Export Development, that is known as APIDA, it is also provides funding. Then Department of Biotechnology, New Delhi, National Horticulture Boards, it is also assist for developing a micropropagation industry in floriculture plants. Then Small Farmers Agribusiness Consortium, that is known as SFAC, related state governments. So different state governments, they focus, they are having different mandates, and they provide the funds. So if some state governments, they are interesting to setting up the industry. So government of UP nowadays, they are providing funds for different institutes, even the private stakeholders. So they are providing a huge amount of funds, and for tissue culture also. And if there are many, suppose this industry may be set up by in a cooperative management or arrangement. If 3 or 4 people, they may gather and they start their business in partnership, and they may apply for a loan from a bank directly. So now the last section of this presentation is the successful examples. So there are lots of industries which are existing in India, and add on this date around 125 plant tissue culture facilities are exist all over the India, and they were certified by National Certificate System for tissue culture plants, that is known as NCS TCP. And these are example of some industries. Out of 125, they are like Shanti Biotech, Sheel Biotech, Sri Mukund Biotech, Pratham Enterprises, AG Biotech Laboratory India Limited, and many others. So, and they are producing different types of plants, and they are assisting other stakeholders and farmers, and many of them, they are involving in the export of the plantlets, even the horticulture and floriculture system. And this is the complete website. So if anyone want to know about these facilities which are operating, so for more details, someone, if you want to know that how they are operating and what are their system, and for what kind of plants they are developing through plant tissue culture, so you may access this website. So dear friends, we have learnt through this lecture the 4 things: that what are the basic concepts of plant tissue culture, second, what are the different parts of the plant tissue culture labs and its organization, and then how one can set up a plant tissue culture unit as an entrepreneurship business, and then finally we have learned or discussed the different successful examples of plant tissue culture units which are existing in India. So, with this, thank you very much, dear friends, and we will meet again in this process, and good luck. Thanks.